Patrick. Welcome, Patrick.
Thank you.
Bum, ba, bum, 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 bum. Wait a couple minutes here, see if we get some more people showing up. There's usually a whole group sitting in a room somewhere, so definitely don't want to start before they get here.
Sounds like a plan.
So how about that Super Bowl? Wasn't Taylor Swift awesome?
Did you have a dog in that fight, or were you rooting
Not
for anyone
really.
in particular?
Not really, but I was kind of pulling for the Chiefs, but
Right.
it was just a fun game. It was a good game.
Yeah, same here actually. I know anyone. After the Niners beat my Packers, I was kind of pro Chiefs. So
What do they say? The enemy of my enemy is my friend.
exactly. So it's funny because the week prior I was rooting for San Fran because I really didn't want Detroit to win. And then 1 week later, I'm like, okay, that's enough roots for San Fran. So
okay, it's 11 o'clock. Why don't we go ahead and get started? I want to start by going over the exam. I don't know if I really have to. You guys did so well on this exam. It was shocking. Shocking coming from Plattsburgh. I mean, I've been teaching undergrads who, I mean, you tell them to study and they don't until the night before, and then they stink on the exam. I'm getting the impression you guys are like studying, which is awesome. So let's look at this exam. Let's go to, there we are. Share, see if we can share this. Okay, stoichiometry of sodium potassium ATPase is obviously 3 sodium out and 2 potassium in, which is the most prevalent phospholipid. That would be phosphocholine. I hope I get these right. It'd be embarrassing if I don't. What causes the depolarization of the EPSP to diminish exponentially? That's your leak potassium channels. Charybdotoxin is potent selective blocker of the Vultr calcium channel or potassium channel. So if you're just blocking some of them, probably what's going to happen. And again, these questions are like hypothesis generation. What do you think? You know, it's kind of critical thinking stuff. So the Vultr potassium channel is important for the repolarization of the cell. So repolarization is going to go slower. If you block them all, you wouldn't get action potentials anymore. It would just screw up the process. But if you have 10% of them, you're probably just going to slow repolarization. You won't have as much overshoot, the hyperpolarization, but you will go back to membrane potential. Okay, or resting potential.
I have a question about that question.
Okay.
If you don't,
sure.
I might have wrote in there, and I'm pretty sure I still got like full credit, but I'm more curious from like me. Would that actually increase the refractory period as well?
Yeah, it would, because the faster you repolarize, the faster those sodium channels go from inactive to closed. So yeah, it would increase the refractory period. We didn't really talk about refractory periods. I guess we should. I guess I can push you a little further now that I know you guys are really paying attention. So yeah, but we didn't talk about refractory periods, but you're right.
All right, thank you.
Okay, what state is the Vultr sodium channel in when the membrane is repolarizing during the action potential? I just said that, inactivated. Okay. The protein provides nucleation point for the synthesis of new microtubule from a paracentriolar material. That would be your gamma tubulin. The grip is a protein that holds this in place. And then Stathmin and Kitanin or other maps that are involved in stabilization or breaking down. This ARP is bound to the side of a microfilament. That is ARP23. WASP and CDC are involved in putting it there, but this is the one that actually binds there. This axonal protein converts the sliding into bending. I think this was on one of our quizzes. That would be Nexin, although I think I said I would accept radial spoke too. I'm not sure, but Nexin is the correct answer here. Okay, another long question. Researchers who studied skeletal muscle contraction developed a model called the skin muscle prep. So basically, this is true too. You would take one muscle cell, dissect a fiber out, hit it with detergents, and get rid of all membranes. So all you have is a sarcomere there, or a series of sarcomeres. So nothing you're going to do membrane related is going to work anymore. What do you need? All you need is calcium to pull that tropomyosin off the myosin binding sites, and ATP to support the cross-bridge cycling. What would you predict with the results from the following experiments? In vitro, a system of magnesium and GTP and free tubulin is established to promote treadmilling. So we're adding at the plus end, subtracting at the minus end. During treadmilling, tubulin monomers are added. Oh, I just said that. When this is established, a solution containing gamma Turks is added, what you would expect to see. So what's going to happen here, because gamma Turks are nucleation points, they're going to start nucleating new microtubules. So the old microtubules will still be treadmilling. The new ones are going to start right at that gamma Turk, and because they're anchored on the minus end by the gamma Turk, they're not going to depolymerize. So they're only going to polymerize. So eventually, I think the ones that aren't already stabilized are just going to go away, and you're only going to see microtubules that are stabilized at one end and polymerizing out. In the second experiment, you're going to add tau. So tau then is a bundling protein. So you're going to see these microtubules bundle together and stick to each other. Okay. Filopodia are parallel arrays. Structural protein at the Z line, alpha actinin. Cytoclazin's a drug. Vinculin is in the focal adhesion, so it's not here, and tau is microtubule associated. This ARP binds the plus end and adds more actin. That would be Mr. Foreman. Okay, concentration of calcium tightly controlled in solids. So this one is not necessarily hypothesis generation, it's just more memorization, but you've got 3 proteins that are decreasing cytosolic calcium and 3 that are increasing it, 3 classes and 2 classes. So to decrease cytosolic calcium, you've got SERCA on the smooth endoplasmic reticulum pumping calcium in there. And then you've got your plasma membrane calcium ATPase and your plasma membrane sodium calcium exchange. And then to get calcium to increase in the cell, you just need channels. So you've got plasma membrane channels, several different types, and you've got 2 different types of smooth ER channels. You've got your ryanodine receptor, which is calcium activated, and the IP3 receptor, which is IP3 activated. As a cell biologist, you're studying mutants of amoeba. You find a strain where the growth of pseudopodia is half the normal rate. This is due to a mutation in one gene, leading to a change in one protein. What protein might this be? So what happened? This is, again, hypothesis generation. So what causes amoeboid movement? You've got depolymerization of monomers in the back end. You've got kind of a contraction with myosin and actin forcing them through to the front, and then adding to the microfilaments at the front end. Some people were talking about ARP2-3 and WASP. Amoeboid movement doesn't do branching. It's just straight gel actin there. So that wasn't really correct. But other than that, a lot of things could be correct. I mean, I gave credit for any of the G proteins, RAC-RO, CDC42, they could be involved here. also, I think mainly what I was thinking was gelsolin or cofilin, which are involved in depolymerization. Or at the other end, you've got, I think the best answer I saw from some people was profilin. Profilin's bringing the monomers into formin to add them. And we saw that one animation where if there's no profilin, it goes a lot slower. So I think that was probably the best answer. But profilin, filamin, there's a lot of different things involved in actin, actin polymerization. Any one of them could be messed up here. So let's see how I did. Okay, well, I guess I did okay, except for the questions I didn't answer. All right, so that was the test. So let me show you real quick how the results came out. A little shocking. Oh, come on, man. All right, let's just do this. See if this works. Sure. Okay, can you see this Excel page?
Yes.
Yep. So in terms of this class, it's just a little shocking to me. We've got it's the 18 and 5 is 23 A's out of I think 30 students or so. So yeah, I'm really pleased with the results. Class average was about 94.8. This includes the bonus points you got for the game. So so that was a good result. Okay. All right. So question about the test? Any questions, or should we just go on and start learning more new stuff? Let's move on. Okay. So let me see if I can pull my, let's try that full screen thing again. Maybe this is the best way to share. Okay, you see my PowerPoint?
Yep.
Okay, good. Let me back up just a tad and talk about receptor kinases, and we'll finish receptor kinases today and possibly move on from here. So anyway, receptor tyrosine kinases, RTKs. These are extremely important in all kinds of things. Mainly, they're going to be involved when you're changing gene transcription, whether it's to cause cell division or cell differentiation. So these are common there. And they are a single transmembrane domain with an extracellular ligand binding site and an intracellular tyrosine kinase. And we always draw it like this, like it's a big long chain. This is more correct, what it actually looks like. And when the hormone binds to the receptor, it will then bind together with another receptor of the same type. All right, and when they bind together, they cross-phosphorylate on tyrosine residues. And each of these tyrosine residues then is a different SH2 binding site, SH2 domain binding site. So a lot of different proteins can be recruited to this activated receptor by these tyrosines. I'll come back. Well, so here we've got STAT, phospholipase C gamma, which we'll talk about today, SHIC, which is an adapter protein, JAX, which we may get to today, GRAB2, which is an adapter protein, the STATs, which we'll talk about, SHIC, and PTP, protein phosphotyrosine phosphatase. Remember, that's our one of our feedback negative regulators. And
Are all those that you just Are all those that you just listed what you're referring to as SH2 domains? Like those are all different types, or is that something different?
These are all examples of different types of SH2 binding domains. So
Okay.
Each of these will bind a different SH2 domain.
Thank you.
And then you've got different SH2 domains on different proteins. Now this is where the term SH2 domain comes from the protein SRC. SRC homology domain, it stands for, and SRC is a kinase that's associated with focal adhesions and other areas too, but that's usually where I think of it, in the focal adhesion. And that SH2 domain then has kind of like a binding site for the phosphotyrosine and for those 3 flanking amino acids. And this is kind of jokey. It's like a plug and play thing. You can literally do this in the lab if you want. You can take an SH2 domain for maybe GFP, right? The green fluorescent protein, stick the SH2 domain on that GFP, and show the cells under fluorescence, and you'll see that it's everywhere in the cell, and then stimulate with an enzyme, with a growth factor, and boom, it'll all move to the membrane. So you really can plug and play it. But in terms of evolution, these things evolved in one protein probably, and then spread to other proteins because they were so useful. The other type of binding domain then is our SH3, which is binding these polyproline regions, and they kind of stick out because they twist and bend when you put a bunch of them together. Okay, so we got SH2 and SH3 domains, and they end up in a lot of different proteins, and this is going to cause that recruitment, bringing things up to the membrane. And when you're recruiting to the membrane, you're bringing enzymes close to their substrates, and that's activating a lot of signal transduction pathways. Here's the one we're most interested in right now, GRB2 SOS. So GRB2 is an adapter protein. It's got an SH2 domain on one side and 2 SH3 domains. And those SH3 domains keep it attached to SOS, which is a GEF, guanine nucleotide exchange factor. So I threw these in here this time. I'm kind of updating my PowerPoints as I repeat myself. So SOS, son of Sevenless. Sevenless was the gene that was deleted in Drosophila when they saw this. So this is a normal eye structure, which has got 7 ommatidia in it. And when they deleted, I believe it was the EGF receptor, they ended up with only 6 ommatidia in the eye. So they called it Sevenless. And then they found that this protein, SOS, was related to that. So they called it son of Sevenless. So Sevenless was basically the receptor, and this was son of Sevenless because it was related to it. That's not important. It's just history, and it's kind of fun. It's an interesting history fact, so I like to throw that out when I can. Okay, so here's what a GEF does. So this would be what SOS is doing. Your G protein in the inactive form has got GDP attached to it. The GEF will then kick GDP off. GTP can then bind to it. And then you got it in the active form. And then it'll go off and bind to some other enzyme to activate it. And GAP is another protein with an SH2 domain that'll be brought up for negative feedback. And it is going to stimulate the GTPase activity of RAS, or of the G protein, sorry, and cause it to break that GTP down into GDP. So what was I going to say? So anyway, this is G protein cycling. And as long as you've got hormone there binding to the receptor, activating or bringing SOS up there, you'll keep activating this RAS, or this G protein. And then once the hormone goes away, this will shut it off. And that's what I wanted to say. It's like GAP is another one of those, what do we call them? Tumor suppressors, that as long as it's there, the cell is okay. But if you lose this, the spontaneous hydrolysis of GTP is so slow that this stays activated, and so that can make the cell become cancerous. So GAP would be a tumor suppressor, and we'll talk about cancer a couple chapters from now, so it's not that important now. Okay, so here's what actually is happening with our RTK mechanism for MAP kinase. So a lot of different pathways can be activated by RTKs. This is one of them. So your receptor binds to, or the hormone binds to receptor, dimerase, autophosphorylate, GRB2 brings up SOS, and that's going to cause RAS to become activated. RAS then, let's go if it's GDP, it picks up GTP. So that's the beginning of the mechanism. And here, this is just showing what some of these other SH2 domain containing proteins can do. This is an adapter. So it's got an SH2 domain here and an SH2 binding domain here. So when it gets brought up, it gets phosphorylated, and then that brings this guy up too, and then that gets turned on. As I say, as we get into the signal transduction stuff, you're going to be like, man, I wish I went into rocket science. It would be so much easier. So here's the whole pathway. This is our kinase cascade over here. Receptor, phosphorylation, GRB2 SOS, activates RAS. RAS then activates RAF, which is a serine threonine kinase, which activates MEK, which is another kinase. And then MEK activates MAP kinase, and MAP kinase goes into the nucleus and phosphorylates transcription factors and causes changes in transcription. Remember, too, that this MAP kinase is often called ERK, extracellular related kinase. So this is called that just because, you know, your extracellular stimulation activates it. So ERK, ERK1, ERK2. Oh boy. And here, another figure from your book. This is showing a lot of the negative feedback pathways. So ERK then can phosphorylate MEK and RAF, turning them off. And it also upregulates the production of the phosphatase that's going to dephosphorylate these proteins. Phos, ETS. So these are transcription factors that are getting activated, and they're going to cause the cell to go into cell division. Cell cycle. We'll talk about that again in the next chapter. Okay, and that brings us to the PI3 kinase mechanism. So this is new today. So this is just another pathway that's activated by RTKs, and it's really, really common and really important. So, and it all has to do with our phosphatidylinositol. We started talking this right in the first lecture, I believe. Phosphatidylinositol then has got this inositol as its polar head group. And when we were talking about actin regulation, we said there are PI kinases that phosphorylated at the 4 and 5 position. So this is PIP2. So we'll see PIP2 again later in this chapter when we talk about phospholipase C. Phospholipase C will break that bond and release IP3. IP3 binds to the IP3 receptor and releases calcium from the membrane. But in this particular mechanism, we're talking about PI3 kinase. So PI3 kinase is going to put a phosphate in the 3 position right here. So now you've got this huge, ugly polar head group here with 3 phosphates sticking out. And that's like a huge sore thumb sticking out, and other proteins are going to come up and bind to that. And just as a sideline here, PTEN is our negative feedback mechanism. Again, PTEN has got SH2 domains, and when you activate the receptors, you bring PTEN up as well as other proteins. So PTEN is going to reverse this by taking that phosphate off. And PTEN is another tumor suppressor. If you lose PTEN, this mechanism doesn't shut off and the cell keeps dividing. So why does PIP3 matter? Here's PIP3. We've got a different domain similar to the really SH3 domain, in that it's going to come up and bind to this PIP3. And this is another kind of plug and play domain, in that it will you can be on a lot of different proteins, and it's going to bring these proteins up to the membrane, and then they're going to be active because then they're going to be near their substrates. So you can see here's the pH domain and the kinase domain that's attached to it. This is going to be AKT, which we'll talk about in a second. Okay, so here's your pH domain over here in living color. You've got arginine, lysine, arginine. These are all positively charged amino acids that are interacting with the phosphates there. And that's why it's being brought up, because you've got this amino acid structure that is specific for it. Oh God, my dog's barking. So I'm debating whether I should pause. He'll stop barking. We'll leave him alone. Okay, so here are 3 examples of enzymes that are going to be brought to the membrane by this: PDK1, PDK2, and AKT. Now, PDK2 is also called mTOR. And again, I'm throwing a little history in here because I find it interesting. mTOR is the mammalian target of rapamycin. Rapamycin is a drug that was found on Easter Island, Rapa Nui. Some scientists went there looking for new antibiotics or whatever, biodiversity they could find there that would be useful for medicines. And they found this drug and they called it rapamycin for Rapa Nui. And it is used as an immune suppressant because it can block immune cell replication. So this is the target of that particular protein. That's why it's called mTOR. And there are 2 different versions of mTOR, 2 different isoforms. One tends to be close to the membrane, and one is a little more cytosolic. I'm not sure. I wrote about that in the notes. I'm not sure if we're going to see that though. And then AKT. AKT is also called protein kinase B, and that's kind of the main target here in this pathway. PDK1 and mTOR are going to phosphorylate AKT, and that's going to further activate it. So then it can, phosphorylate targets downstream. Now here's our downstream target. So this is called the destruction complex, and we're going to see this in a couple different pathways and some unrelated pathways, but it's important in all these pathways. This is a complex of a number of proteins. And if you do a Google search and do Google images or even Wikipedia, you'll find there are a lot of different proteins that are involved in this destruction complex. And it changes based on the cell. Like I've got WTX here. And later I'll show you a destruction complex with a different protein there. It just kind of, there's a lot of variability here between cells. So we've got these 3 proteins, APC, axin, and WTX, which are kind of like scaffolding proteins, especially axin. And they're holding GSK3 beta in the right conformation so that it can beta-catenin. Beta-catenin is a transcription factor. So if it is unphosphorylated, it will be a transcription factor. But when it gets phosphorylated, that targets it for ubiquitination and the whole ubiquitin proteasome pathway, which we haven't talked about. At this point, just think, okay, phosphorylated, degraded. That's all that really matters. When we get into apoptosis, I think that's where we're going to talk about this pathway more. But for now, that's it. Okay, and we can tell how important this protein is, GSK3 beta, glycogen synthase kinase beta. We know why this is important because this protein, APC, is Adenomatous polyposis coli. So it's named for a cancer that forms when it's absent. So that's colon cancer. WTX, Wilms tumor protein. So when that's absent, there are kidney tumors. So this complex is really important, and you know that because if you mess with it, you get tumors, basically. We'll also see later when I show the destruction complex that the ubiquitinating enzyme is part of this complex as well. And another kinase, casein kinase, is also part of this complex in some cases, maybe in all cases. But anyway, bottom line, destruction complex, constantly phosphorylating beta-catenin, kind of like a tonic phosphorylation, causing it to be degraded, keeping the levels low. Okay, so what happens when so this is this is stimulated. So when you have, let's make this bigger. This is the PI3 kinase pathway. When the hormone is around, it binds to the receptor. The receptor then recruits PI3 kinase with its SH2 domain. Here's your SH2 binding domain. And then now it's near the membrane, it's going to phosphorylate PIP2 into PIP3. So you get a bunch of PIP3 in the membrane. That brings mTOR, protein kinase B or AKT, and PDK2 up to the membrane, and they're going to phosphorylate AKT. AKT then becomes active, and it's going to phosphorylate GSK3 beta, and that's going to block GSK3 beta. So normally GSK3 beta is blocking beta-catenin. When this comes along, it blocks GSK3 beta. Now beta-catenin builds up in the cell, goes into the nucleus, and is going to bind to a protein called TCF-LF. And we'll see that in a later slide, so don't worry about it. And that's going to become a heterodimer and a transcription activator. So there's your pathway. PI3 kinase makes PIP3, bringing these proteins up. AKT then phosphorylates GSK3 beta. It's also going to phosphorylate BAD. Let's see. Let me look real quick. Am I talking about that in the next? Yeah, I'm talking about that in the next slide. So any questions about this before I move on? Okay. So next slide. Oh, by the way, I can't see your chat. So if somebody lights up the chat, maybe someone could tell me about it because I won't see it while I'm here. Okay, so where are we? So this complicated slide, this is from your book. Not crazy about it, but it does tell us something important. And it is that this pathway, this PI3 kinase AKT3 pathway is about causing the cell to go into division, cell division, okay? GSK3 beta is blocking beta-catenin. When beta-catenin is around, we're going to get c-Myc and cyclin D1. These mean cell division. So these are going to cause the cell to go into S phase and then get ready to go into mitosis. So that's just what we were talking about there. But this is also going to be important for a lot of other things that are involved in getting that cell ready to divide. If you think about it, you need more than just turn the cell on. You're going to need a bigger cell, and you're going to have to activate a whole bunch of stuff and inhibit some stuff. So here, AKT is also blocking glycogen synthase, which is which is changing metabolism here. We'll see in it. This pathway is important in the insulin mechanism. We'll see that next. But anyway, it's changing metabolism, increasing metabolism, so you get more energy to build the cell bigger. Another thing you really, really need to do when you're going to cell division is block apoptosis. So BAD is a pro-apoptotic BCL2 protein, which we'll talk about in chapter 11. BIM is a BCL2 protein. Then you got Fas ligand, and I just fixed this in your notes because I had it wrong when I uploaded. But Fas ligand is the ligand that's going to activate the death receptor that causes apoptosis. So we will talk about that a couple chapters from now, so I don't want to talk about too much. But basically, this pathway here is blocking apoptosis. AKT is also going to upregulate the activity of MDM2. And MDM2 is a protein that inhibits P53. So P53 is involved in, it's kind of the switch that tells the cell that it's got a lot of DNA damage. And if it does, it'll go into apoptosis. So this pathway is going to, and again, I'll talk about that more in chapter 11, but this pathway is going to upregulate MDM2, which is going to downregulate P53, so you're going to get less apoptosis. And then the third kind of pathway we're seeing here is TSC23, which is another kinase, and that's going to activate our mTOR raptor, which is mTOR2. mTOR1 is the one up here at the membrane. That's mTOR Richter. And so mTOR raptor then is going to phosphorylate S6 kinase and this ugly named thing here. And this is going to activate E1F4E. So EF14E is one of those transcription, it's a general transcription factor. It's, I believe, anyway, I could be wrong. I'm pretty sure it's a general transcription factor. So we've got specific transcription factors like beta-catenin that turn on, that bind to enhancers, but you also have these general ones that are going to be binding to the to the promoter region and bringing in the RNA transcriptase. So they're increasing transcription of genes in general. And then S6 kinase is going to phosphorylate this protein, which is associated with the ribosomes and increases the rate of translation. So bottom line, what's going on here? AKT is activating TSC1/2, which is activating mTOR raptor, which is stimulating transcription and translation. We're really upregulating the cell's ability to make proteins. And that's what you're going to need in the G2 phase of the cell cycle to make the cell bigger so it's ready for mitosis. So just to kind of review, if the cell is going to go into division, it needs to activate division, and it also needs to inhibit apoptosis and stimulate transcription and translation. So all this gets upregulated. Any questions? Comments? Okay, so next we'll talk about the insulin, the way insulin works, which is, I think it's really a neat mechanism. And I throw it in here because it's very similar to the PI3 kinase mechanism. It's almost the same, except for the end targets. So the insulin receptor is not one protein, it's a heterodimer of alpha and beta subunits, and they bind together and they form the full receptor. And then you've got your PDK1, PDK2, or mTOR, AKT, PI3 kinase is part of this. One new protein is IRS1. So IRS1 is got SH2 domains, and it will be attracted to the activated receptor, and then it will bring PI3 kinase up. The other thing we got going on over here is GSK3 beta, again, our destruction complex. And GSK3 beta is tonically phosphorylating glycogen synthase. So glycogen synthase, as the name suggests, is going to make glycogen from glucose. So in an unstimulated cell, GSK3 beta is blocking the action of glycogen synthase. And that way, if there isn't a lot of sugar out in the blood, and so there's no insulin being released by the body, then you're not making glycogen. Okay, you're allowing the sugar in cells to be glucose, and it can go out into the blood and keep your brain working. Basically, I don't think mine's working right now, but in general, keep brains working. Anyway, so you've had a meal, your blood glucose increases, your pancreas releases insulin. Insulin binds to the receptor. The receptor then brings IRS up. IRS brings PI3 kinase up. So then it starts making PIP3. And that PIP3 then attracts PDK1, AKT, and mTOR. But just like the last mechanism, PDK1 and PDK2 phosphorylate AKT. And now AKT is going to do two things. It's going to come down and phosphorylate GSK3 beta like it did in the other mechanism and inhibit it. And that's going to allow glycogen synthase to be active. And the other thing it's going to do is phosphorylate a protein on on these vesicles. So in general, you should think about how membrane proteins are regulated. And I'll talk about this probably the last thing in this chapter, although it's not in your book, but we should talk about it. Any membrane protein is going to be in the membrane, but you're also going to have a pool of vesicles slightly below the membrane that'll have those proteins in. And that way, if a cell needs more in the membrane, it puts them into the membrane. If it's got too many, it pulls them out. And it's kind of like this buffer zone of membrane proteins that can be inserted or taken out of the membrane as needed. So in this pool of vesicles here, we're talking like a liver cell or something, you've got GLUT4, which is a glucose transporter, passive facilitated transport. And so there's a protein on this vesicle that's called, what's it called? It's called AS160. When AKT gets activated, it's going to phosphorylate AS160, and the vesicle then is going to fuse with the membrane, and then you end up with more glucose transporters in the membrane. So glucose transporter then will bring this excess glucose in, and when it comes in, glycogen synthase is on, and you're going to make glycogen. So I actually have an animation of this that I made back in the day in Flash, and I need to update it so it can be seen in HTML5. I hope to get that up this week. We'll see. But anyway, bottom line, the mechanism is insulin, IRS1, autophosphorylation or cross-phosphorylation. IRS1 comes up, PI3 kinase comes up, phosphorylating the PIP2 into PIP3. Your kinases then come up to the membrane, PDK1, PDK2 phosphorylate AKT. AKT then phosphorylates AS160, and your transporters go in the membrane, and it inhibits GSK3 beta by phosphorylating, and then glycogen synthase is ready. And bada boom, bada bing, we're making glycogen, and we're decreasing blood glucose. Life is good. Okay. Questions? Probably not. Okay. Just shout them out if you got questions. So next, oops, is this next? Yes, this is next. Okay. Okay, so here's another pathway activated the PI3 kinase mechanism. Let me back up and show you how I presaged this. You like the word presage? I do. Okay, so you can see here phospholipase C gamma, another protein with an SH2 domain with its SH2 binding domain. So this is another pathway that's going to get activated here. And this is a pathway I keep alluding to and never talking about specifically, because I've been saving it for the G protein coupled receptors, but here it is anyway. Ooh, this is wrong. This is PKC gamma. This should be PLC gamma. I'm going to have to fix that. So this is phospholipase C gamma, and it's got an SH2 domain, binds the receptor, and then it's going to break PIP2 down into IP3 and diacylglycerol. Diacylglycerol activate protein kinase C, which is another enzyme that can be associated with cell division. And IP3 then binds to the IP3 receptor on the ER and releases calcium. So you get calcium release here. Okay, let me pause for about 5 minutes because I think my dog has to go to the bathroom. I'm gonna have to let him out. He's yipping. I'll be right back. Okay, sorry about that. Gus's stools have been running a little loose lately, so could have been a real mess if I didn't let him out. All right, phospholipase C. I got to fix this slide. It's definitely wrong. Okay, so here's a figure from your book again. This shows the 3 different pathways we talked about that are associated with receptor tyrosine kinases. Here's your RAS MAP kinase pathway. And then you've got your PI3 kinase pathway, and then your phospholipase C pathway. Okay. And I think my figures are better, but that's me. All right, we move on. Okay. Next pathway. This is also a receptor tyrosine kinase, but it's different from the ones we just talked about because the receptor is actually separate from the kinase. So the kinase is not a part of the same protein. And we've got 4 different proteins that could be associated here with these receptors. Well, let me just say again, JAK-STAT, this is a JAK-STAT pathway. I actually had a student, her last name was JAK-STAT once. It really made for some funny discussion during cell biology. Okay, so important in embryo development, especially important for immune cell function, because your cytokines are going to go through JAK-STATs. And it's constant, it's common in blood cancers for that reason. And it's also common in mammary cancers. So we've got 3 different enzyme kinases that can attach these proteins. 3 of them are JAKs, JAK 1, 2, and 3, and JAK stands for Janus kinase. Although back in the day, people used to call it just another kinase because there's so many damn kinases involved here. But you've got JAKs 1, 2, and 3, and TYK2, tyrosine kinase 2. So those are separate. So anyway, bottom up, this is the coolest part about the stat part of it, of this mechanism. But this is almost the same as what we've seen already. The receptor, the hormone comes, receptors clump together, cross phosphorylation, activating the kinase domains here. Now they're going to phosphorylate the receptor, making SH2 binding domains. And your stats, which are going to be the transcription factors, they've got both an SH2 domain and an SH2 binding domain. So they've got both. And so this SH2 binding domain SH2 domain attracts it to the membrane. So you get them binding there to the receptor. The kinases then phosphorylate the stats, and then they come away, and now you've got heterodimerization through these SH2 domains. Kind of beautiful, I think. And then it just goes in the nucleus and acts as a transcription factor. There may be some other co-activators that it needs to bind to, but that's the bottom line. Let me look at my notes. Yeah, it moves into the nucleus there, and that's your transcription factor. So that's it for the JAK-STAT pathway. It's pretty simple, really, compared to the MAP kinase pathway. Now our next pathway, just another pathway. By the way, this chapter is complicated because there are a lot of pathways, and you need to know them all. So yeah, here we go. So these are receptor kinases, but they're a little different in that they're going to phosphorylate on serine and threonine instead of tyrosine, which is more common, right? Most kinases are going to phosphorylate on serine or threonine. And these receptors are really important in embryo development. I don't know if you've had developmental biology, but we've got, well, actually I wrote them down here. You've got TGF beta, which is somewhat important in development. BMP4 is like vital in development. And then you've got nodals in Xenopus, they're called XNR, and then activin and VEG1. So these are all hormones that are going to activate serine threonine kinase receptors. And then what they do is activate the SMADs. SMAD. So the name SMAD is something mothers against decaplegic. I don't know. Decaplegic is another hormone that is going to activate these types of receptors. So SMAD is something, I don't know what the first word is, mothers against decaplegic. But anyway, you've got 2 different types of SMAD. You've got a receptor SMAD and a cellular SMAD. There's only one, as far as I know, only one cellular SMAD, and that's SMAD4. Receptor SMAD, though, you've got basically 2 different classes of them. You've got SMADs 1, 5, and 8, and then you've got SMADs 2 and 3. And they're kind of going to be activating separate things. You definitely see this in development where BMP4 is going to activate SMADs, I believe, 2 and 3, and then the nodals are going to activate 1, 5, and 8. And they're going to turn on different genes, and that's going to be a difference between ventral and dorsal in the developing embryo. That's not important for this class. Bottom line being, we've got different SMADs that are going to be activated to activate different downstream genes. So the other thing that's kind of weird about these receptors compared to receptor tyrosine kinases is you've got two different receptors. You've got the type 1 receptor and the type 2 receptor, and they're both important. The type 1 receptor actually binds the hormone. The type 2 receptor is going to bind to the type 1 when the hormone is present. So here's how the mechanism goes. You've got all this in the unstimulated state, and then when the hormone is around, the receptors clump together. Type 1s bind the hormone, type 2s bind the type 1, and then they cross-phosphorylate each other, further stimulating their kinase activity. Once that happens, the RSMADs are going to be phosphorylated, and that's going to activate them, and the RSMAD will then bind to the SMAD4, making a heterotrimer, and that will go into the nucleus and cause changes in transcription. Yeah, I guess that's all I have for this. Okay, so another pathway, another kinase, and we're always happy when we have another kinase. All right, any questions, comments? All right, I want to do one more pathway if we're okay with that. It's similar to the, it has a common target as the PI3 kinase pathway. So it's kind of relevant to what we talked about today. But I don't want to go any further than this Wnt pathway. So this is going to be the last thing we talk about today. So getting back to that destruction complex we were talking about, one protein is called APC, Adenomatous Polyposis coli, and that is involved in this, in the formation of new cells in your large intestine. So in your large intestine, you've got these crypts, and the crypts are involved in making new cells because you know those cells on the surface are going to die and slough off and constantly be replaced. So down at the bottom of this crypt, you've got stem cells constantly dividing, and it's Wnt signaling that's going to cause these to divide. And so they're going to be, first they divide, and then they get pushed up. As you get new cells here, the old cells get pushed up, and you still have Wnt signaling all the way through here until this point. And at this point, Wnt signaling stops, and the cells are going to differentiate from stem cells into epithelial cells. And then they'll move up and out onto the surface, and they live their life and they die like we all do. And then they get replaced by others like we all do. So anyway, this production of new epithelial cells in the large intestine is all about Wnt signaling. And here's an example of what happens when it's not signaling right. You start getting a tumor down the bottom here, and then that fills up the crypt and then starts to balloon up out of the crypt. And that's what your polyp is. And when we get into cancer, we'll discuss why this is the significant event. I mean, polyps into the colon are not that big a deal. You know, you should be getting your colonoscopy every 10 years. If they see some, they'll cut them off, and then they'll have you get a colonoscopy sooner. But if these cells go through this way, they're going across this basement membrane and they're starting to metastasize into the body. That's when you're in big trouble in terms of cancer. So anyway, why am I telling you all this? That one protein I told in the destruction complex, APC, is named for this particular disease because if you lack APC, then you're going to have a lot of Wnt signaling, a lot of polyps. And if you can Google images search Adenomatous Polyposis coli and look at a colon, it is all polyps. The whole thing is polyps. There's no normal skin in the or epithelium in the colon. Anyway. Okay, so here is the Wnt pathway. I believe this. Nope. I got this from the internet. This particular figure. Stretch this out there. Okay, so here's the destruction complex. And now we've got some different proteins added into this figure that I alluded to earlier. You've got APC here, GSK3 beta, and axin. So you've got those three here. You've also got CK1, which is casein kinase. And so GSK3 beta and casein kinase are going to tonically phosphorylate beta catenin. So they're constantly phosphorylating that. And this beta TRCP, this is a ubiquitinating enzyme. So this is going to add this protein ubiquitin to beta catenin. And then your proteasome is here as part of the complex, which is breaking down beta catenin. And again, we haven't talked about this ubiquitin pathway. We will. What's most important to know here is that GSK3 beta and casein kinase are phosphorylating beta catenin, causing it to be broken down. So same thing as we saw in the PI3 kinase pathway, we're tonically breaking down beta catenin. Now, up here at the membrane, we've got two receptors. Frizzled is the main receptor, and LRP is kind of the co-receptor. And then here, this guy disheveled is another protein that's going to be important. That's DVL here, disheveled. And you can kind of guess where these names came from, right? They're Drosophila names again. They had a mutant that looked kind of disheveled, so they called it a disheveled mutant. They had another mutant that looked kind of frizzled, so they called it frizzled. So that's where these names come from. But anyway, what's going to happen here in this mechanism is if you've got Wnt around, Wnt is going to bind to the two receptors, the frizzled and the LRP. That's going to bring disheveled up, and that brings axin off the complex, or at the very least, it's bringing the whole complex up here, and that's turning off GSK3 beta. So the once GSK3 beta is turned off, beta catenin is no longer phosphorylated, and it goes into the nucleus, binds to TCF-LF, and it starts to causes cell division. So that's your pathway. Pretty more proteins to memorize, but kind of a short pathway. Wnt binds to the receptor and the co-receptor. That brings disheveled up, which brings axin up, and that stops the inhibition of GSK3 beta. That inhibits GSK3 beta, causing beta catenin to build up in the cell. That is what we call the canonical Wnt pathway. Canonical because this is normally what you see. I guess it's canonical because it was the first pathway that was figured out for Wnt, but later people realized there were other pathways that Wnt can activate. Here's your non-canonical calcium pathway, which is going to sound, again, like something else we saw with the receptor tyrosine kinases. It's going to bind to a G protein coupled receptor, which is going to activate phospholipase C, breaking down IP3, releasing calcium. I'm not going to talk much about this because we'll talk about this later in this chapter when we do G protein coupled receptors. But again, calcium release, protein kinase C activation. This is a little different, CAM2 kinase. We may talk about that later, but I'm not really sure. Your book talks about CAM2 kinase a bit, but this is your non-canonical calcium pathway for Wnt. And then you've got another non-canonical pathway, which is kind of interesting in my opinion, and this is the planar cell polarity pathway. So it's called planar cell polarity because this is going to be kind of like one of those what am I looking for? I talked about this in chapter 3, when there's a chemical and the cell is chasing it, going towards it in one direction, right? What did I call that? Chemosensing? Oh God, my brain is blanking on the word. But basically, in chapter 3, I talked about how if you've got a gradient of a specific chemical, cells can crawl towards it. So what we're talking about here is activating cell crawling. So with this pathway, again, you've got frizzled and LRP here as receptors, and disheveled gets brought up, and that activates DAMM. DAAM stands for disheveled associated activator of morphogenesis. Morphogenesis, changing cell shape. So DAAM is going to activate Rho, And then we've got another G protein here that's going to activate RAC. So RAC Rho, remember, we remember them from chapter 3 and cell crawling. So RAC is going to activate ROCK, which is Rho activated cell kinase. I have it written down here. Rho associated kinase. I don't know where the C comes from. So, and then this is going to remodel your actin microfilaments into stress fibers, and RAC then is going to activate JNK, June N-terminal kinase, and that's going to cause changes in transcription. But also RAC, we remember, is involved in remodeling actin microfilaments into lamellipodia. So basically what we're doing is activating directed movement. So the cell is going to move in the direction that wind is coming from. Okay. That's about as far as I want to go today. I only have one more slide, but I want to say hedgehog because we've gone through a lot of pathways already. Another pathway I might throw out there because I really like is the hippo pathway used in really early embryo development. We'll see. Maybe I'll do it. Maybe I won't. But anyway, what the hell are you talking about, Slish? Shut the hell up. Sometimes I'm like sitting in a room by myself and it's like, just like I'm sitting in a room by myself. I'm just going to yak away. I don't know if any of this makes any sense, but it's fun to talk. Everyone likes talking. Can you turn your cameras on real quick so we can feel like we're not alone? Okay. Okay, some of you have come up. That is Matthew. Can I call you Matt? Okay, I'm not going to. I just want to know if I can. David Roy. And there's Brittany. Hey, Brittany. And Patrick. All right. Any questions or comments? We had enough for one day? All right. Let's call it a day. And we'll meet back here next Monday. Don't forget, we're going to have a quiz this weekend on everything we've covered since the last exam. 10 questions, usually not that hard. Maybe I'm going to start stepping up my quizzes a little bit here. You guys are doing too well. I can't give everybody an A. Can I? Would it be okay if I gave everybody an A? All right, you're all good kids. All right, talk to you later, guys.